This is the Lee Lab headed by Dr. Ching-Sheng Lee, and we work with HIV, specifically looking at transmission projects as well as a lot of different materials and methods and model organisms. So a lot that is done here focuses on using novel breeds, types of mice, specifically the BLT mice, bone marrow, liver, and thymus. Transfer mice that are humanized, but my project in particular focuses on SIV, simian immunodeficiency virus, and looking at earliest detection with rectal transmission as the form of transmission. And we use RNA-scope in situ hybridization to actually look in non-lymphoid tissues, so lung, liver, and brain, just sort of looking to see when that first event of the SIV transmission happens in that non-lymphoid tissue.
I'm at the Buffett Cancer Research Center at UNMC, and I'm in Dr. Dada's lab, and we are studying the advanced prostate cancer. So specifically, I'm studying an adhesion molecule called NR-CAM, which is specific for prostate cancer. We've come to learn that as prostate cancer advances, it turns into a more neuroendocrine-like cancer. And with this neuroendocrine-like cancer, there is a neuroendocrine-specific adhesion molecule, NR-CAM, which we suspect helps in the aggressive proliferation of this cancer. And so we're trying to find ways that if we knock down this adhesion molecule, is that going to help slow down the proliferation of this cancer? In the lab, I've learned to run Western blots to look at protein concentrations and just to understand if we do a experiment where we're trying to knock down NR-CAM, we want to see that knockdown as well in the protein level to make sure that what we're trying to knock down is actually being knocked down and it isn't something else or it's not actually being knocked down. And we've done IHC, which is immunohistone chemistry, just to understand we get actual patient samples and we want to look at different components of their samples, patients who do have prostate cancer.
So I'm working with Dr. Michael Herman here at UNL in the biology department. It's working with this model organism called C. rhabditis elegans. They're a 1 millimeter long transparent worm that you can see very easily under the microscope, and they sort of, wiggle around very elegantly too, which is how they got their name elegans. And yeah, they're amazing for studying a lot of biological processes because they're easy to maintain in the lab. They're transparent, so we can see a lot of processes that are going on inside of them, like in their organs and cells. Currently, we're using it to study host pathogenic response mechanisms against this emerging new antibiotic-resistant bacteria, Stenotrophomonas maltophilia. I've been working on this project that studies specific proteins that are involved in the immune response against the bacteria. And I've been using some really cool technologies to do that, including gene editing to make those proteins glow when they express. So it's been a lot of troubleshooting using gene editing technologies, but also just working with the worms in general. It's been a good experience. You know, there's a lot of science behind all the genetics that I do, but at the end of the day, I'm just making glow-in-the-dark worms, and it's just so fun.
Getting the experience and being a part of the NE-INBRE program, I feel like has really given me a step ahead of where I would have been if I wasn't in the program, and just a cool idea of what undergraduate research looks like and kind of being able to get that experience early on while I'm still in undergraduate school and not transitioning into graduate school and not having that experience, I feel like has really helped me a lot.